Hi, this is Mikaela and you're watching my vlog.
So the materials that we need for this 3D bacterial cell is cartolina, scissors, pencil, paintbrush, paint, yarn, glue, glue gun, glue sticks, beads, fuzzy wires, and construction papers. First thing we have to do is trace the shape of the bacterial cell in a cartoon. Next is cut out the part you trace with scissors. Next, use the cutout to trace the other shapes. Next, I glued them all together using a glue gun. Next, I glued the top part that will serve as our cell wall. I cut out some cartolina for covering the sides that will serve as our capsule. Next, I painted the cell wall red paint. Next, I used the yarn that will serve as our nucleoid. Next, I glued the fuzzy wire surrounding the nucleoid that will serve as our plasma membrane. Next, I glued the small beads that will serve as our ribosomes. I cut out a small part of the fuzzy wire and glued it that will serve as our plasmid. Lastly, I glued the fuzzy wire surrounding the structure that will serve as our pili and flagellum. So this is it. This is the final output of our 3D bacterial cell.
As we all know, bacterial cells are prokaryotic cells, lacking well-defined nuclei and membrane-bound organelles, and with chromosomes composed of a single closed DNA circle. They come in many shapes and sizes from minute spheres, cylinders, and spiral threads, to flagellated rods and filamentous chains. They are found practically everywhere on Earth and live in some of the most unusual and seemingly inhospitable places. This pink part right here is called the capsule. Capsules play a number of roles, but the most important are to keep the bacterium from drying out and to protect it from the phagocytosis by larger microorganisms. This red part right here is called the cell wall. The wall gives the cell its shape and surrounds the plasma membrane, protecting it from the environment. It also helps to anchor appendages like the pili and flagellum, which originate in the plasma membrane and protrude through the wall to the outside. This green part right here is the cytoplasm. The cytoplasm is where the functions for cell growth, metabolism, and replication are carried out. It is a gel-like matrix composed of water, enzymes, nutrients, waste, and gases, and contains cell structures such as ribosomes, chromosome, and plasmids. This violet part that surrounds the cytoplasm is called the plasma membrane. The plasma membrane encloses the interior of the bacterium, regulating the flow of materials in and out of the cell. This is a structural trait bacteria share with all other living cells, a barrier that allows them to selectively interact with their environment. This yellow part of yarn right here is called the nucleoid. The nucleoid is a region of cytoplasm where the chromosomal DNA is located. It is not a membrane-bound nucleus, but simply an area of the cytoplasm where the strands of DNA are found. These little beads right here is called the ribosomes. The ribosomes are microscopic factories found in all cells, including bacteria. They translate the genetic code from the molecular language of nucleic acid to that of amino acids, the building blocks of proteins. This small strand right here located inside the cell is called the plasmids. Plasmids are small extrachromosomal genetic structures carried by many strains of bacteria. Like the chromosome, plasmids are made of circular pieces of DNA. Unlike the chromosome, they are not involved in reproduction. These small strands right here is called the pili. Many species of bacteria have pili, small hair-like projections emerging from the outside cell surface. These outgrowths assist the bacteria in attaching to other cells and surfaces, such as teeth, intestines, and rocks. This long strand right here is called the flagellum. The flagellum are hair-like structures that provide a means of locomotion for those bacteria that have them. They can be found at either or both ends of a bacterium or all over its surface.